The second speaker today will be Professor Svante Pääbo, who is one of the founders of paleogenetics, a discipline which is a relatively new discipline, which is related to the genetic analysis of our early ancestors, the hominids. Svante has made pioneering discoveries in the field. He was the first one who, with his collaborators, described in 2010 in Science the DNA of the Neanderthalian. His book, The Neanderthalian Human, has been published in several languages in tens of thousands of copies. More recently, he has made another very seminal and pioneering discovery. He discovered a distant relative of the Neanderthalians, if I understand correctly, the Denisov man, from a little piece of bone found in a Russian cave, a Denisov. And his recent analysis shows how these two distant relatives of us Homo sapiens have influenced the development of our genome by intermarrying between these three different distant relatives of hominids. He has several important prizes, including the very famous and distinguished Leibniz Prize, and he is member of different academies, including the Royal Society of London, the Académie des Sciences of France, and of course the Swedish Academy, since he is Swedish. And ladies and gentlemen, in the interest of time, allow me to ask our second speaker, Professor Svante Pääbo, to continue. Please keep your questions, and after Professor Svante Pääbo's lecture, we will have a bio break, followed by a question and answer session, when you can ask questions to both Francis and Svante.
Okay, so, dear Balázs, dear Sydney, dear friends, it's really a great pleasure to be back here in Singapore again. It is indeed a very dynamic and exciting place. So, what I then wanted to do today is just start out by reminding you about what you all know, that the human genome is a rather big place, around 3 billion base pairs. If we compare two genomes, we have a difference about every 1,000 nucleotides or so. So we have in the order of 3 million differences between two genomes. So it's quite a lot of information in order to reconstruct history, genetic history. So if you now do this for humans on a worldwide scale, what you will find, you also know, I think, is that most variation among humans exists in Africa. And although, of course, there are many, many more people living outside Africa today than inside Africa, we have less variation outside Africa. And not only that, most DNA sequences you find outside Africa have rather close relatives inside Africa. But there is a component of the variation there in Africa that have no close relatives outside Africa. So this pattern then is a sort of genetic basis for a recent African origin model, in addition to the fossils that Francis then talked about. So the inference out of this is that modern humans evolved in Africa, accumulated genetic variation there, and a part of that variation left Africa and colonized the rest of the world. With genetic tricks, you can also estimate approximately when that exodus out of Africa happened by looking at over how long distances variants are associated with each other, the extent of linkage disequilibrium in the genome, and it's rather recently in evolutionary terms, yet less than 100,000 years or so. So maybe I should actually come back in a month from now and give a talk here. But there is one problem, if you like, with this recent African origin model. And that is the fact that we know that there were other forms of humans around since much, much further back. In particular then, in Western Eurasia, the Neanderthals, which exist there since 300,000–400,000 years ago, and in Asia, less well understood, sort of groups of now extinct hominins. So my laboratory is fascinated since 15–20 years with particularly the Neanderthals. This is a Neanderthal skeleton on the left compared to a modern human skeleton. And these Neanderthals then, as I said, appear in Western Eurasia somewhere 300,000–400,000 years ago and live there until in the order of 40,000 years ago when they become extinct. And why are we so interested in Neanderthals? Well, I think there are at least two reasons for that. One is the fact that Neanderthals are the closest evolutionary relatives to all present-day humans. So if we should sort of define ourselves as a biological group, it's really them we should compare ourselves to. Another interesting question is also sort of how are we related to them? When did we share a common ancestor with them? Did we interact with them genetically? Did we mix with each other and so on? So that last question, if modern humans mixed with Neanderthals, and it's sort of a controversial theme in anthropology for decades actually, with all kinds of ideas going from total replacement, saying modern humans come out of Africa, replace Neanderthals with no mixture whatsoever, 0% contribution from Neanderthals to present-day people, in a sliding scale to sort of total continuity, saying Neanderthals are the direct ancestors of present-day Europeans. So our first chance to address this with genetic means came in the early 90s when we got permission to sample a Neanderthal fossil, not just any Neanderthal, but a type specimen that was found in 1856 and gave its name to this group of fossils sent from the place where it was found, in Neandertal, in Germany. From the upper left arm, at that time we needed rather much material to do this type of work. We could profit from, by then already, almost 20 years of experience in avoiding contamination with DNA from present-day humans by lots of working in sort of clean room conditions, sterilizing all your equipment, etc., etc. And we focused on the mitochondrial genome for the simple fact that there are many copies of the mitochondrial genome per cell, so it's a bit bigger chance that some fragments of mitochondrial DNA will survive. And with the technology of the time, PCR, then with great effort, we constructed a particularly valuable part of the mitochondrial genome of the Neanderthal. And if you then estimate the phylogenetic tree for the mitochondrial genome of Neanderthals, comparing it to present-day humans, what you find is what was already known, that the mitochondrial genome of present-day humans go back to a common ancestor between 100–200,000 years ago. The Neanderthal mitochondrial genome goes back much further, about 500,000 years or a little more, to common ancestors shared with present-day humans. Since then, we and others have looked at many other mitochondrial genomes of Neanderthals. They all fall outside the variation together with this one here. So it's clear that in this scale of things, for the mitochondrial genome, it's total replacement. There is no one running around today with a mitochondrial genome from a Neanderthal. But it's also quite clear that the mitochondrial genome is a rather boring part of our genome, if you like. It's very small, it's maternal inherited as one unit, so it's a big stochastic component in how it is passed on to the next generation. Any woman who has no daughters will not pass on her mitochondrial genome. So the big picture of our history is of course to be found in the nuclear genome. And I think I'm sort of on the published record in some article somewhere, in some newspaper 10 years ago now, saying we will probably never sequence the Neanderthal genome, there's too little of it there, the nuclear genome is too degraded, etc., etc. And I think it just goes to say that you should never make negative predictions like that, because you're generally overtaken, at least in biology, by sort of history, and history in the form of technology. And what came around that I hadn't really anticipated was high-throughput DNA sequencing. So what you could start to do is just extract all the DNA from a fossil, make a library, and sequence all the DNA that's there. And then use the human genome that was by then sequenced to sort of see which part of the DNA, which fragments of DNA might come from the Neanderthal genome. First place where this worked was in southern Europe, this place in Jacquet. This little fossil, almost 30,000 years old, is part of it here. And the first thing that you then notice if you look at the DNA in there is that it's degraded to short fragments. So 50, 60 base pair long fragments, hardly anything longer than 200. And the vast majority of the fragments are actually not at all from the Neanderthal. So the very best Neanderthal fossils we have something like 3-4% of the DNA from the Neanderthal, and all the rest from bacteria, fungi, other organisms that have grown in the bone. So we were then very lucky to be funded for 5 years to improve the methods with which we extract DNA from fossils, convert it to sequencing libraries, and isolate the fragments of interest in there. So we got a lot better on that, and could then start sort of approaching the Neanderthal nuclear genome. We also screened a lot of our archaeological sites, a lot of bones, to find the ones with the highest proportion of endogenous DNA. Focused on 3 bones from 3 different individuals from that site in Croatia, in Jacquet. Sequenced a bit over 1 billion DNA molecules from them, but at the time was quite a lot. Nowadays it's not that much. The vast majority of this was then fungal or bacterial, but you could then map it to the human genome, taking into account that there are particular chemically induced errors in the sequences, particular cytosines appearing as thymidines towards the ends of the molecules. And we could then map these things until we then, in 2010, had about 1 full genomic coverage, saying each position is statistically seen once. So that means there's still quite a lot of the genome not seen. So we had a little over half the genome that we had seen at that point. And we could begin to ask questions. And one of the first questions was this, then, has there been interbreeding with present-day humans when they came out of Africa and met Neanderthals in Western Eurasia? We addressed that with many different methods. The sort of most direct, intuitive one is then to say that if Neanderthals had mixed with modern humans, say, in Europe, we would expect Europeans today to share more alleles, more genetic variants with Neanderthals than people in Africa, where Neanderthals had never been. So there's no reason to assume that there would be any Neanderthal contribution to Africans. So we had the Neanderthal genome. We sequenced 5 genomes from different parts of the world in order to have exactly the same error profiles in these genomes, because we did them totally in parallel in our lab. And then did a very simple test. Just take 2 genomes, and to test this idea, we start with 2 Africans that should have no contribution from Neanderthals. We find all positions in the genome of these 2 individuals differ from each other. And we then take the Neanderthal genome and say, how often does a Neanderthal match this African individual or that African individual? And it should be 50-50, right? Because there's no reason to assume that one African is closer to Neanderthal than another African. And indeed, statistically speaking, that's what you see. So we then take 1 European individual and 1 African individual and do the same test. And to my surprise at the time, actually, we found statistically significantly more matching with the European individual than the African individual, suggesting that it could be a contribution from Neanderthals into the ancestral Europeans. Even more surprising when we took a Chinese individual and an African individual, we again see more matching to the Neanderthal. Although most people would think there have never been Neanderthals in China, although one might perhaps discuss that. But when we take someone from Papua New Guinea, where for sure Neanderthals have never been, we again see the same pattern. So there's sort of model that came out of that has since largely been proven right, was to say that when modern humans come out of Africa, presumably they pass by the Middle East. We know there are Neanderthals there. So if these early modern humans that early come out of Africa, mix with Neanderthals, and then become the ancestors of everybody else outside Africa, they can sort of carry with them this Neanderthal contribution to the entire rest of the world, also to parts then when Neanderthals are not existing. To the extent then that if your roots are outside Africa, somewhere between 1% and 2% of your genome come from Neanderthals. So when we then published that in 2010, there was quite quickly a lot of follow-up by other groups who published scientific papers. That was quite fun. What I was also sort of surprised by the time then was how big an interest there was from the general public in this. So almost immediately did we start getting lots of emails and letters from people who self-identified as Neanderthals and wanted to send us blood samples and hair samples. And I started noticing a pattern in this correspondence, that it was primarily men who wrote to us. They said they were Neanderthals, and very few women who wrote to us and claimed they are Neanderthals. So I sort of presented this to my group as my research. I count emails, and they are of course hypercritical of anything I say. So they said this is just ascertainment, men are somehow more interested in genetics, so they will write to you, women are not. But that is not true, because I went back to my emails and counted again, and there were quite a few women who write and say they're married to Neanderthals. And very few men who claim they're married to Neanderthal women. So that's of course genetically quite interesting. But there are other things going on too. So particularly we started working with colleagues in Russia who excavate in Siberia, particularly this site in southern Siberia on the border to China and Mongolia, the Nisova Cave. And in 2010 they found a toe bone there, a little toe bone, and by then we had developed much more sensitive ways of doing DNA libraries from ancient DNA. Methods that rely on not using the double-stranded DNA to add adapters to the ends and sequences, but start out by separating the two strands, by denaturing the DNA, and by single-strand ligation, add an adapter, isolate this on a solid support, synthesize the other strand, so that each double-stranded molecule has two chances to make it into the library. And that turned out to be quite important for this ancient DNA, because it often carries chemical modifications that make it impossible to replicate the sequence of DNA. But that's often confined, say, to one strand, so you then have the other strands that you can go into the library. So with this and some other tricks, and using this toe bone, which turned out to come from Neanderthal, we were then able to go from a one-fold coverage of the genome to about 50-fold. So we now have a very high-quality Neanderthal genome. So we can then use this genome to, say, look in present-day people, illustrated on one chromosome here, or different people from 1,000 genomes data, to say what fragments come in these individuals from Europe here, from Neanderthals. And you will see that different individuals often carry different fragments from the Neanderthals. So we all carry often different parts. So adding up to something like 1 or 2% in every individual. But what you then can ask is how much of the Neanderthal genome exists today still in people walking around on two legs. If we sort of jump from individual to individual, how much can we put together? That's indicated in blue here. And that's still a bit uncertain, but it adds up to at least 40%, or probably half of the Neanderthal genome is still around today in different people. But that was not the only thing that they found there in the Nisera cave, as we already heard in the introduction. They also found back in 2008 a tiny little bone fragment. And I don't know if there are many archaeologists in the audience, I should perhaps be cautious, but sort of Western archaeologists have a tendency to talk down about Russian archaeologists and say, you know, they excavate like we did 40 years ago, they're just looking, finding the big things. But I think realizing that this little fragment may be of hominin origin was actually extremely skilled. So we got this little fragment, actually didn't pay attention to it for almost a year in the lab, and then extracted DNA from it. And we're able to reconstruct a good genome from it, of high quality, and we're very surprised to find that it was not a modern human, it was not a Neanderthal, but something else that go back quite far back in time to common ancestors shared with Neanderthals. So if we say that the deepest divergence among present-day human populations is in the order of 100,000 years, the common ancestral population shared between Neanderthals and Denisovans is about 4 times deeper than that, something like in the order of 400,000 years. So we call these individuals Denisovans, or this group Denisovans, just as after this site where they've been found. So Neanderthals are called Neanderthals after the Neanderthal site, and these guys are called Denisovans after the Nisera cave. You can ask for them as for the Neanderthals, have they contributed to present-day people? And indeed they have. So if you look, anywhere you look in mainland Asia, you have a small contribution of Denisovan DNA, less than 1%, in the order of 0.2, 0.3%. But if you look in Oceania, in Aboriginal Australians, Papua New Guinea, you have up to 5% of contribution from Neanderthals. So what you can also then see in these genomes is that they have very little variation within their groups. So in present-day people, you can see that the heterozygosity, the sort of amount of variation between the 2 chromosomes you carry, is higher in Africa than in non-Africans, and these Neanderthals and Denisovans have again smaller variations, suggesting that they live in small groups over a long time. You can then, with a good genome, start looking at gene flow not only from these guys into present-day humans, but also among them. And we find lots of evidence of that. So we find this gene flow, I've already said, from Neanderthals into the ancestors of all non-Africans today. There's probably additional gene flow from Neanderthals into the ancestors of people in Asia, because Asians have something like 20% more Neanderthal contribution than Europeans. They said 1%, 1.2% approximately. There is a contribution from Neanderthals into Denisovans of small magnitude. There is this contribution from Denisovans to people all over Asia. And in addition, a larger contribution to people in Oceania then. And quite interestingly, the Denisovan genome had an old component not present in Neanderthals. So from something that diverged more than 1 million years ago or so, that contributed to Denisovans, a few percent. So it's very tempting to say that this is something like Homo erectus in Asia or something like that, but we don't know that. And there is recent evidence also that some early modern ancestors from Africa contributed to Neanderthals earlier. So this is a very complicated picture. It's probably going to get more complicated in the future. I think the take-home lesson is simply that humans have always mixed with each other, at least to some extent. We don't find massive mixtures, but we find sort of small contributions in almost every direction. We were reminded about that recently from a site in Romania this site where cavers found this mandible in 2010, I think. It's 40,000 years ago, and it looks like a modern human. So it's one of the earliest modern humans that we know of in Europe. So it lived at a time when there were Neanderthals still around. So we were very interested in saying, have this individual mixed with Neanderthals or not? So we just look at one chromosome here and indicate in some modern humans here in color fragments coming from Neanderthals. And then there's a question, is this guy on top here, has that guy mixed with Neanderthals or not? And indeed it has, and massively more than these other modern humans. And in fact, the parts that are in black here, we have no information, so it seems like over half of this chromosome is solidly of Neanderthal origin. That, of course, suggests that there was a Neanderthal ancestor close in the family tree of this individual. So there are 7 such regions in the genome here where large parts of chromosomes come from Neanderthals. So you can model that and say that it shows that in the family tree of this 40,000-year-old individuals, 6, 5, or 4 generations back was there a Neanderthal ancestor. So we sort of almost caught someone in the act, if you like, of mixing with Neanderthals. And perhaps this indicates that this happened more commonly than we thought in the past. So the future will have to tell that. We sort of know that when modern humans spread, they mixed a couple of times with Neanderthals, also locally in Europe, probably mixed with other forms, say in China, and probably also in Africa. I wouldn't be surprised if that will appear in the future. So what has happened is that we've clearly sort of rejected the total replacement model. There is a contribution from these earlier forms of humans to present-day people, but it's of rather small magnitude. Maximum, say, 7-8% or so in Melanesians. We like to sort of call this model, say, leaky replacement or so. The big picture is one of replacement. When people come out of Africa, there's a small contribution from other forms of humans. So for the last part of the talk then, I want to sort of address the question, well, does this mean anything? What role does it play in the biology of present-day people? So have Neanderthals made contributions, genetic contributions of any functional importance to present-day people? So what you can then do is we can go over the entire genome here, say in Europeans, and plot the frequency in each place of Neanderthal contributions in the population. And you will find in some cases, mostly it's 1 or 2 or 3% of people carrying Neanderthal DNA, but there are things that have risen to 50, 60, 70%. Some of these are shared with Asians, some are unique to Asians, some are unique to Europeans. So those are things that then seems to have been of importance and risen in frequency. So what are those things? And we're beginning to learn that. There's almost monthly papers appearing now looking at these things. So one of these things in high frequency is, for example, a region containing this gene, which is a lipid transporter, that have turned out to be a risk factor for type 2 diabetes in Asians. But this variant is quite frequent, say 25% of Native Americans or people in China carry this variant, and it confers additional risk to get type 2 diabetes. If you then plot the tree of the risk alleles and the protective alleles, the Neanderthal falls right in. It's a big chunk of 40,000 base pairs that come from Neanderthals into people in Asia. So why would something that confers a risk to disease today have risen to high frequency? I think there's good reason to sort of assume that variants that increase the risk for diabetes today, when we eat almost too much during our entire lives, actually may be protective in a situation of starvation. So this may be some Neanderthal adaptation to starvation that was advantageous in the past, and now have this sort of side effects. in the present-day population. So there are many other examples from Neanderthals. I just want to bring a few examples from from Denisovans also. So Denisovans have also contributed to present-day people. So particularly in Tibet, people in Tibet are adapted to living at high altitude where there's little oxygen in the air. It was already known that one major adaptation to that is a variant in a gene called PAS1 that had to do with how much oxygen you can take up without increasing your hematocrit, your red blood cell count. So a group at Berkeley showed that this variant exists in 80% allele frequency in Tibet, and that variant comes over from Denisovans into the ancestors of Tibetans. So it's sort of quite fascinating that we may not have had so high population densities in the Tibet had we not had this contribution from Denisovans. There is also what seems to be an adaptation to cold climate that one has identified in Greenland. There is this variant of a gene, it's probably this gene here that has to do with the development of brown fat that generates heat when you uncouple oxidative phosphorylation in the mitochondria, that is fixed, almost fixed in Greenlanders, and occur also in other populations. That variant turns out to, these variants here, is closely related to Denisovans and further away from Neanderthals, and this is the typical variants that occur in the rest of the world. So there seems to be this high adaptation to life. So sort of Denisovans, if we take a step back, Denisovans are sort of an enigmatic group. We know bone remains of them so far only from this site in southern Siberia. We know that they have contributed up to 5% of the genomes in Oceania. They've also contributed to this adaptation in Tibet to life at high altitudes. There is some probably cold adaptation, but they have contributed to people in the subarctic. So a big question is, where did they really live? And that's something that is still, what was their distribution in the past? And I think that will come from studying fossils in China, for example. But since a couple of months, I'm also very optimistic that there will be progress on this, because we have recent sort of developments in our lab where we have sampled from excavations, not bones, but just sediments. So instead of studying bones from excavation, we extract DNA just from sediments that look like this, and can actually retrieve at least mitochondrial DNA from it. So from a number of sites in Western Eurasia, we have then shown what animals were present at these sites in the past, and have also been able to show if Neanderthals or modern humans were present in the caves, also where you find no bones remaining of them. So the hope is that one can now go to archaeological sites where one just finds, say, stone tools and other remains of human activity, and see where Denisovans, for example, have lived across Asia. That's something that I hope will happen in the next few years. But I also want to sort of continue this sort of functional things here, saying there are also variants that seem to have been so important that they've been contributed multiple times, both from Neanderthals and from Denisovans to present-day people. So there are, for example, toll-like receptors, which are cell surface receptors important in innate immunity. And these variants have this typical distribution of occurring outside Africa, only a little bit inside Africa, associated with gene flow back into Africa. And if you look at these variants for these toll-like receptors, they've been contributed from Neanderthals and also from Denisovans independently of them. And if you look at people today that are homozygote for these Neanderthal or Denisovan variants, they express higher levels on their white blood cells of toll-like receptors compared to the ones that don't carry these variants or are heterozygous. If you look in association studies of people with these variants, they are then have increased resistance to Helicobacter pylori infections, so the type of infections that give you ulcers, but they also have increased risk of allergic diseases. There's sort of many such things are appearing. So many things that have been positively selected that one now finds in the genome. Many of them have to do actually with immunity and with skin and pigmentation for some reason. So there's sort of a picture emerging where these modern humans come out of Africa, they meet these groups, Neanderthals and Denisovans, that have lived for hundreds of thousands of years in this environment in Eurasia, mix a little bit with them, pick up some variants that are advantageous, and they rise to high frequencies. It's something one could call adaptive introgression or so. So, but we should sort of not forget that most of the variants that one finds are not sort of beneficial that one finds today. They sort of have to do with different diseases, things such as hypercoagulation, many things with skin diseases, also things that have to do with the brain, such as depression and so on. There's a recent interesting study showing that if you look at the expression of genes contributed from Neanderthals, where you can then look at the allelic imbalance, so look at people that express the modern human variant and the archaic variant, there's a tendency in the brain particularly to express the Neanderthal variant at a lower level, and also in testes, there's some evidence of that. There's often a tendency to plot it would be 50-50 of the two alleles, but the Neanderthal alleles tend to be expressed at lower levels. So there seems to be, particularly in the brain, and some selection against expressing these variants. And that sort of brings us to the last part of the talk here, where I wanted to discuss what was not contributed, if you like, from Neanderthals. And that, I think, may be particularly interesting, actually, for the future. So what you can now do is you can go over the whole genome, chromosome by chromosome, plot in red here, contributions in Europeans from Neanderthals, in green in Asians from Neanderthals. And then you can look for regions of the genome where you statistically would expect the contribution from Neanderthals, but you see none, or very, very little. So these are things we call deserts in the genome, where we do not see Neanderthal contribution, as if we select against it. And why are we so interested in that? Well, it's because we imagine that there might be functions hiding in those regions that are really important for being a fully modern human. So we're interested in things that changed here in the genome since we separated from Neanderthals and spread to everyone, or almost everyone. And why are we interested in these things? Well, I think that modern humans are, after all, quite special in some ways. For me, as a sort of geneticist, if someone shows me stone tools that the Neanderthals made in the beginning of their history, say 400,000 years ago, and in the end of their history, 40,000 years ago, they look pretty similar to me. An expert can explain to me that there are differences there, but to me they are just stone tools. But if you then look at modern humans and look at modern human technology 100,000 years ago and today, over a much, much shorter time, I needn't be an expert to say that technology has changed dramatically over this time. So there is something very special with how our culture and technology changes. So also things as figurative art that really depicts something, comes only with modern humans. And only modern humans, of course, start spreading across open water, colonizing the whole planet. So we think that there's something special with modern human sociality or cognition. And a dream is then for the next 5 years or so that one might be able to find some of the basis for that and these types of changes here. And we can list all these changes now because we have the Neanderthal genome. We can make a catalog of them. And that list of things is not very long. It's in the order of 30,000 changes in the genome. that are fixed in present-day humans. Everybody have them, and Neanderthals look like the apes. So, just to give you a feeling for how we might begin to think about these things, let us focus on a little subset of them, the ones that change amino acids in proteins. That's just 96 changes in total. And they occur in 87 proteins. So you can look at them and say which ones are important. Also the ones that may affect expression. So we have a bias, of course, in what is important. We think it should have to do with the brain and cognition, so development or function of the brain. So one way then to look for these 96 proteins to say, are they overexpressed? Are they particularly expressed, these 87 proteins, in some part of the developing brain? And it turns out that against the control of proteins with silent changes that don't change amino acids, it's in the proliferative zone of this epithelium that make the cortex during development. But this relies on very few observations, right? It's just 87 proteins in total. There's like 6 proteins that are responsible for this. But quite surprisingly, 3 of these proteins actually have to do with the spindle and the kinetic core, so the machinery that sort of pull the chromosomes apart during cell division. So that was really surprising to me. I thought that cell division would be something that would be so conserved that it would not have changed recently in human evolution. One speculated, of course, when we found this and say, well, we know that mitosis during brain development here, the cleavage plane is very important, where if your stem cells will make a neuron and a stem cell, or 2 stem cells that will go on proliferate. So maybe something with that. But that was then just speculation. But we now begin to have tools to address this, of course. And the tools are iPS cells, induced pluripotent stem cells, that we can differentiate in vitro to different types of neurons and astrocytes, but also to these cerebral organoids, as you know. So sort of small mini brains, if you like, that mimic early development of the brain. So we have a group studying this in our institute, comparing first these organoids to fetal brain, showing that these organoids mimic gene expression, say at the single cell level, very well from the fetuses. We make organoids from chimpanzees, so we can compare the early brain development in apes and humans, and they turn out to be very, very similar to each other. But there are some interesting differences. So this is work by postdoc Nora Bermudez in a group that we work closely together with on this, studying the division of the apical progenitors here in this epithelium. So if we now look in time-lapse microscopy on the mitoses, you will see the chromosomes here, and you will see them lining out in this metaphase plate and then be pulled apart. And something that we found was that this metaphase in humans compared to chimpanzees and also orangutans is longer. So this period where the chromosomes line up and are pulled apart are longer, and that's specific to these neuronal precursor cells, doesn't occur in other mitoses we study. So that, of course, sort of gives some justification to this speculation. And it's sort of tempting to speculate that that might have to do with these changes in these proteins involved in spindle and kinetic cores. So what we then, of course, want to do now is to introduce these changes into the iPS cells and study these effects in the organoids, and also introduce these changes into the mouse and study mouse brain development. And if I have 4 or 5 minutes more, yes, then I just sort of want to illustrate a bit that one might be able in the future to also use mouse models for this type of work. And that then hasn't to do with these genes that we are just beginning to get into the mouse now, but another gene that we studied since 10 years. So that is a gene found by Tony Monaco in Oxford some years ago, where they studied a family with a language and speech problem. And that leads over a little bit to the next sort of talks perhaps about the evolution of language. So this family had a severe language and speech problem. They were able to find a gene by linkage in this. It turned out to encode the transcription factor, FOXP2. Interestingly, we found that this transcription factor is very conserved among mammals, but it carries 2 amino acid changes in humans relative to the apes. That's unique to humans, close to each other in the protein, in a part of the protein that hasn't got any clear function that is defined. So we were very interested, of course, if these changes occurred in Neanderthals and Denisovans also, or if they might be unique to modern humans. So when we started, these are these 2 positions, and when we then started getting fragments from the Neanderthal and Denisovans, we looked at these positions, and they turned out to be identical to present-day humans. So, in other words, these 2 changes happened early on the lineage here. We share them with the Neanderthals, but they might, of course, still be interesting, because they are then located in a gene, affect the gene, that has something to do with language and speech. So, since this protein is so conserved from mouse to human, there's only one other change here that is conservative, we hope. What you can then do is to humanize a mouse. You simply introduce these 2 changes in the mouse FOXP2 gene. And you then have a mouse that from its endogenous FOXP2 gene essentially makes a human protein. And you have the problem that you then want to study language ability. It's like a mouse, so you try to speak to the mouse and it will not answer. Probably the lifespan is not long enough to really acquire language. It's a problem. But you have an animal model that you can now study. So for example, you can look at sort of neuronal development and compare then the humanized mice to wild-type litter males, so the homozygous humanized mice. And you find that they make longer neurons, so longer dendrites and axons. So there is something going on with the neurons. And those changes are particularly in the cortical basal ganglia circuits, so layer 5-6 in the motor cortex, in the basal ganglia, but not, for example, in the cerebellum that also express FOXP2. Since these circuits have to do with motor learning, it's sort of then tempting to say maybe it has something to do with motor learning, and that stimulated a previous graduate student to go to Andrea, MIT as a postdoc with the mice and study motor learning in the mice. So these are experiments where the mouse had to go in a maze and had to find chocolate milk, which the mice like very much. And these are then experiments where one gives some light signal, for example, say on the left side, and the mouse after a while learned that I should turn towards the light here to get my chocolate milk. And there is no difference between the humanized mice and the wild-type mice in how quickly they learn that this light is important for learning where the chocolate milk is. But it then comes a time when you can take away the light. If you gave the signal to this group of mice always to the left, the mice will after a while learn, I just go to the left. And you can take away the light that is an internalized this knowledge, I just turn left. And that switch happens in 7 to 8 days in the humanized mice, and in 11 to 12 days in the wild-type litter males. So they sort of automate a muscle sort of motor activity much quicker. So the neurobiologists will call this switch from declarative to procedural learning. And when they explain this to me, they say, for example, think about when you learn to bike as a kid. When you begin to bike, you think about what you do, and you're very bad at it, right? You say, oh, I kick to that, I should actually counterintuitively steer to that side, and it's very bad. But it comes a point where suddenly you stop thinking about what you do. And that is a time when you're suddenly very good at biking, when you don't think anymore. And that is the switch from declarative procedural learning. It goes together with the switch of activity from the medial striatum to the lateral striatum. So we went back to our mice and looked at synaptic plasticity in the lateral striatum, which is increased in the humanized mice, whereas when we look in the medial striatum, it's, if anything, decreased instead, or there's actually no significant difference. So this sort of fits this hypothesis that this has to do with sort of proceduralization of motor activity. So I think this illustrates that what we've learned from the mouse is that these two amino acid substitutions that are unique to humans affect something in cortical basal ganglia circuits that make proceduralization of motor activities more efficient. And it's very tempting then to say that the most sophisticated proceduralization, automated motor activity we do as humans is articulate speech. We need millisecond control between vocal cords, tongues, lips to produce articulate speech. It takes us a few years to learn as kids, and it's something that is very unique among primates to humans. It's sort of tempting to say this has to do with articulation. I think this is very encouraging to suggest that we might be able to use not only organoids, things like that, but also actually mouse models for some of these things. So we're in the process now of putting in regulatory changes in a FOXP2 on top of those changes that are specific to modern humans relative to Neanderthals to see what that makes to what happens then. So it's encouraging that several of these genes that affect sort of have changes in genes that affect brain development or brain function and might be able to sort of clarify in the next few years what we do. So to end then, I hope that I convinced you that if you're interested in human evolution, particularly recent human evolution, it's very useful to have the Neanderthal genome because you can then focus on things that happen here or happen back here. You can time changes and perhaps focus in particularly on the changes that are unique to modern humans. But I think the real excitement now for the next few years will be to test these changes functionally. So, and the way to do that, I think, is to use iPS cells, editing iPS cells, and look at the effects, but also to modify and humanize mice and study them. So, there are many people who are involved in what I talked about, many archaeologists, paleontologists, too many people to mention. I mentioned just one person who developed that ultra-sensitive library method that made it possible to make high coverage Neanderthal penicillin genomes that we would otherwise not have, Matthias Meyer. Many people have helped analyzing the data. I mentioned particularly David Reich at the Broad, Monteslacken at Berkeley, Janet Kelso from South Africa, who's heading all the bioinformatics at our place, and Kai Kruger, and the people who work on the functional aspects in the Institute, particularly the group in Leipzig, Barbara Treutlein, who works on the organoids, single-cell genomics, and Wieland Puttner's group at the Max Planck Institute in Dresden, who help us a lot with the mouse analysis. And with that, thank you for your attention.